Okay, we'll continue with the live attenuated vaccines. And these vaccines, as we already know, were the first to be developed by Louis Pasteur. The microorganisms, they can be attenuated so that they lose their ability to cause disease, but they retain their activity to cause immunity, and they retain their capacity to grow in the host. And most commonly, attenuation is achieved by growing the organism in prolonged conditions, particularly bacteria in adverse conditions like the development of BCG, Bacillus calmette-guerin, which is a vaccine for tuberculosis, and this was actually developed by growing the strain of Mycobacterium bovis in increasing concentration of bile salts for a period of almost 13 years, and after 13 years the strain got attenuated and it could be used as a vaccine. Those bacteria, the mycobacteria, in case of mycobacteria, these bacteria was the bacteria that was used was not Mycobacterium tuberculosis but Mycobacterium bovis. In case of viruses, the attenuation can be achieved by growing the virus in a cell that is cell type that is not normal for the virus. For example, oral polio vaccine. Oral polio vaccine was developed because of culturing of polio virus in monkey kidney epithelial cell. Then the virus got attenuated, and then this was used as a vaccine. The other examples of live attenuated vaccines are measles, mumps, rubella, etc. Okay, then the advantages of these vaccines are that they can grow in host, they can produce long-term memory, because in these cases the organism grows transiently in the host, and therefore the exposure of the host immune system to the foreign antigens is for a longer period of time. That is why they are more potent in developing immunity, and there are no booster doses required for such kind of vaccines. And this is a good point when you consider third world countries, because according to data, almost 30% of people don't turn back for the booster doses in third world countries, in developing countries. So disadvantages are that the attenuated forms, they can revert back to the virulent form. This can happen in polio. A 1 in 2.4 million chances is there that a dose of polio vaccine can generate paralytic polio. There are certain complications associated with the measles vaccine as well. People develop encephalitis like symptoms, but these cannot be taken as general rules and cannot be taken as excuses to not get vaccinated, because the benefit is much larger as compared to their disadvantages. So we'll come to the other class of vaccines, which are inactivated or killed vaccines. Inactivation or killing of the pathogen is done in this case, and inactivation can be carried out by heat or by chemical means. Generally heat is not preferred, because proteins, they tend to denature in presence of heat. Heat denatures the protein, so epitope structure can be lost, and therefore no immunity could be generated. So most commonly chemicals are to be used, and these chemicals are used. Chemicals such as formaldehyde is most commonly used. These kind of vaccines require booster doses. There are risks that are associated, is that pathogen has to be grown in large numbers prior to inactivation. So those individuals who are working in those environment, who are working for inactivation of the vaccines, they are prone to risk and they could develop diseases. Some of the pathogen may not get killed, and this can cause serious problems. Pertussis vaccines, flu vaccines, most of these vaccines are these vaccines. I'll not go into DNA vaccines right now. I'll go to subunit vaccines. So in the previous two vaccines, you see that we used the whole organism as a vaccine, but later on it was found out that why to use a whole organism, why can't we use parts of the organism? So this led to the development of subunit vaccines. So purified macromolecules that are derived from pathogens, they can be used and as vaccines, and this was proved. And one such kind of vaccine is a toxoid vaccine. Some bacteria, are pathogenic because of the exotoxins that they produce, like tetanus, Clostridium tetani, which produces tetanus, and for diphtheria as well. So in these cases, the exotoxins have been purified, they have been inactivated with formaldehyde, and the inactivated toxoid is now used as immunizing agent or vaccine. So the immune system neutralizes the toxin whenever an infection occurs. These type of vaccines are for diphtheria and as well as tetanus. Now this is also another interesting story, which is conjugate vaccines, or these are vaccines. I'll first come to the vaccines which are polysaccharide vaccines. So in case of Streptococcus pneumoniae, the virulence is due to the surface coat, which is made up of polysaccharides, and people have used these polysaccharides, purified them and used them as vaccine, and this provides immunity. So polysaccharides from Streptococcus pneumoniae are conjugated. They are not sufficient enough to generate immunity. They are conjugated with some kind of protein, like in case of Haemophilus influenzae, the polysaccharides are conjugated with tetanus toxoid, and this proves to be highly immunogenic, and this results in immunity to Haemophilus. Same is true for Streptococcus pneumoniae. In case of Streptococcus pneumoniae, we have two types of vaccines. One is simple capsular polysaccharides, another is a conjugate vaccine where some toxoid is conjugated to it, and it increases the immunity manifolds. Then we'll come to adjuvants. Adjuvants, like in the previous case, in case of conjugate vaccines, the toxoid, the tetanus toxoid, can be considered as an adjuvant, which actually primes the immune system and it enhances the immune system's response, and therefore this can be used. Yeah, this is like, we'll go to DNA vaccines now. Yeah, there is another class of vaccines which is which also falls under the category of subunit vaccines, that is recombinant protein vaccines. In recombinant protein vaccines, the vaccine that is used is hepatitis B vaccine. This actually hepatitis B vaccine comprises of the hepatitis B surface antigen protein, which is present on the surface of hepatitis B virus, and this protein in itself is sufficient enough to generate immunity. Nowadays it's not purified from the virus itself. We have cloned the gene into yeast like Saccharomyces and Pichia, and these organisms are used for heterologous expression. The protein is purified and that protein is used as vaccine. So no need of culturing the organism, no need of handling the organism, rather than we can use directly the subunit of the organism. Maybe in this case for hepatitis B, we use the protein that can provide immunity against the viruses. I'll talk about DNA vaccines in subsequent lectures.